Okay. Okay, we were discussing about structure propensities of. Can you hear me, Aisha Siddiqa? Please unmute your mic and let me know. Can you
Yes,
hear
sir, I
me?
can
Okay.
hear.
Okay. Thank you. Mute your mic. So we were discussing about structure propensities of the specific amino acids for three-dimensional structure prediction. There are two ways to analyze the view of and the significance of the propensity of specific amino acids, and it also controls the folding of protein and the specific amino acid selective pressure in the specific region where it will be. And then we have Chou Fasman method, and we were here. Chou Fasman algorithm. Then we have this propensity of alpha helical structures, propensity of beta sheets, and propensity of turn based on observed propensities in proteins of known structure. Here is the table. We have the partial table, not all the amino acids. I hope you guys, everyone can see this table, that Chou Fasman propensities of glutamate, methionine, alanine, valine, isoleucine, tyrosine, proline, and glycine. Sorry, glycine. So P alpha means propensity of alpha helical structure. P beta is propensity of beta pleated sheet, and P T is, or P turn is the propensity of a turn. Kashif Salim. Do we have Kashif Salim with us? Please unmute your mic, Kashif Salim. Kashif Salim. Okay. Abu Bakr. Do we have Abu Bakr with us? Aruj. Aruj is in your class. Shanaziz.
Yes, sir. Yes.
Okay, Shanaziz, here we have glutamate, methionine, alanine. Be with me, okay? So glutamate, methionine, alanine, valine, isoleucine, tyrosine, proline, and glycine. And here we have the specific propensities of these amino acids for alpha helical sheet, for beta pleated sheet, or alpha helices, beta pleated sheet, and turns. So you can see that that this table have the derived values of X-ray crystallographic and NMR structures solved and placed in PDB or any other database. So it was calculated that if there is a glutamate, there is 1.51% chances, or the value propensity value is 1.51, and beta pleated sheet has 0.37 and turn has 0.74. So which one has the biggest value, Shanaziz? Kashif Salim.
सर सर मैं मैसेज कर रहा था क्लास को कि सब क्लास में आ जाओ.
Okay, you can mute your mic. Do we have Kashif Salim?
सर काशिफ सलीम नहीं क्लास में.
Okay. Aisha Siddiqa, please unmute your mic.
सर वो बच्चे कह रहे हैं
Yes.
सेम लिंक से ज्वाइन नहीं हो रहा. मैं उन्हें गाइड कर दूं.
Yes, sir.
बेटा अभी ये देखें, ग्लूटामेट जो है, ग्लूटामिक एसिड है, इसकी म्यूट योर माइक, एवरीवन म्यूट योर माइक. जस्ट आयशा सदीका, एक्सेप्ट आयशा सदीका, एवरीवन म्यूट योर माइक.
Yes, sir.
बेटा आपने हैंड्स फ्री लगाई हुई है?
Sir,
Glutamic acid has the value 1.51, that is the alpha propensity of specific amino acid.
yes, sir. Sir, हैंड फ्री अभी लगाई है.
सर आपकी आवाज नहीं आ रही.
Kashif Slim, please unmute your mic. Can you hear me? Kashif Slim.
Yes, sir.
ओके बेटा,
Yes,
मैंने
sir.
आपकी सारी क्लास को लॉक कर दिया है और अभी सिर्फ आप मेरे साथ रहोगे और ये लेक्चर कंप्लीट होगा। उसके बाद सोचा जाएगा डिपार्टमेंट के साथ डिस्कस करके कि आप लोगों की ऑनलाइन क्लास ली जाए या सिर्फ लेक्चर रिकॉर्ड करके भेज दिया जाए, क्योंकि आपकी क्लास को एथिक्स नहीं है ऑनलाइन क्लास लेने के। एनी हॉबी विद मी चाउ फैसमैन प्रोपेंसिटीज वी ऑलरेडी
Sure.
डिस्कस, जिसमें आपको मैं बता चुका हूँ कि एक्सरे क्रिस्टलोग्राफिक स्ट्रक्चर्स और एनएमआर के जरिए से हमने ये सारे के सारे वैल्यूज निकाल ली थी, राइट? और काशिफ सलीम, बी विद मी. So that now we have these
Okay.
values, and here we have glutamic acid 1.51, 0.37 and 0.74. So which one has the biggest number?
Sir, 1.51.
1.51. So there is how many chances of alpha helix at the place of glutamic acid instead of beta and turn? Because we have 1.51 and it's the biggest value. Same with the methionine
Yes.
and alanine. For valine, isoleucine and tyrosine, we have the biggest value in beta sheets,
Yellow tables.
yellow color,
Yes, sir.
and in
Beta.
beta sheet. And after that we have the values of proline and glycine in turns, right? So
Yes,
here,
sir.
so from this table we can derive that which amino acid will place in which category, like in alpha helix, in beta sheet, or in turn, right? So did you get the
Yes,
point?
sir.
It's very easy.
Yes, sir.
They just drive the values from X-ray crystallographic structures and NMR and just put the values in a table, and now they just tally the table for the specific amino acids. I hope you got the point. It's not a big concept. It's a very easy and small concept.
Yes, sir.
You are a good student.
Yes,
So
sir.
after that we have Chou Fasman method. A prediction is made for each type of structure for each amino acid, right? So we have
Yes, sir.
to predict every amino acid that where it lie. It will be in alpha helix, it will be in beta sheet, it will be in turn, in loop. Okay, now the result has the ambiguity. Why we have ambiguity in the result sometimes? Because sometimes we have same propensities for two amino acids. Like suppose here we have suppose if we will ignore this turn, you are with me, Kashif Slim?
Yes, sir.
Okay, just have a look. If we just ignore the turn, this column, we have to ignore this column, right? Now we have a
Yes, sir.
proline and we have 0.75
Mm.
value as a alpha helix and
Alpha.
0.55
Yes, sir.
values in beta sheet. So now it is ambiguity.
Beta.
It's a
Beta sheets.
very is yes, it's a very close you can competition. So sometimes
Yes, sir.
these types of values happen and just make the results ambiguous. After that, we have Chou Fasman method. The calculation rules are somewhat ad hoc. The question is, what is ad hoc? Did you listen about the ad hoc contracts?
No, sir.
Okay, sometimes we have ad hoc contract in which that starts, that is a very preliminary concept. Contract starts first time. That is known as ad hoc. Like आपने कभी सुना हो, वो
Yes.
कहते नहीं ad hoc पे job कर रहा है ये. So it is like the first one. So calculation rules
Yes,
are
sir.
somewhat ad hoc. Example methods for helix is, sorry, okay, example method for helix is search for nucleating region where 4 out of 6 amino acids have p alpha greater than 1.03. Extend until 4 consecutive amino acids have an average propensity of alpha is smaller than 1. If region is at least 6 amino acid long, has an average propensity alpha is greater than 1.03 and average propensity alpha is greater than average propensity beta, consider region to be helix. So these three rules will be followed to calculate to calculate the amino acid. It will be in the alpha helix. If there are 6 amino acids and out of those 6 amino acids, 4 amino acids has collectively the value more than 1.05. Yes, man, you have a question?
Sir, in every table we have P alpha less than 1.03 or in this table only.
No, we have to like here we have search for nucleating region where 4 out of 6 amino acids. So if you will
Yes.
plus the values of 4 amino acids, it should be more than 1.03. Suppose one amino acid has value 0.10, 0.20, 0.30, 0.10. If you will combine all
Yes.
these values, it will be less than 1. So there will be no alpha helix. So if the values of the
Okay, sir.
specific amino acids will be more than 1.03, then you will consider that 4 amino acid in the alpha helix. Extend until 4 consecutive amino acids have an average P alpha of greater smaller than 1. And the last concept is,
Okay.
if region is at least 6 amino acid long, you have 6 amino acid in a region and has an average P alpha greater than 1.03, and average P alpha is greater than average P beta, then it will also consider as alpha. So in next example, the concept will be more clear, like scan the peptide. You have a peptide, or what is peptide?
सर
Kashif Slim.
सर पेप्टाइड सर मुझे concept याद था लेकिन still सर मुझे
Okay, what is
याद नहीं आ
protein?
रहा
Protein, what is protein?
सर amino combination of amino acids sir.
Combination of amino acids. So in peptide we have about 20 to 30 amino acids usually. Then we have polypeptide,
Okay, sir.
peptide, and protein.
Polypeptide chain. Okay, sir, I remember, sir.
Okay, that's good. Good.
Polypeptide chain, sir.
Yes, so in peptide we have almost 20 to 30 amino acids. Suppose you have a peptide of 20 amino acids, and where 3 out of 5 contiguous residues have propensity of beta is greater than 100. If you have 3 out of 5 amino acids and you combine the values, or you plus the values, or you add the values of these 3 amino acids, the values, if the values are more than 100, then you will consider it as a beta sheet. These residues nucleate beta strands. Okay, I just explained it. So these residues nucleate beta strands, extend these in both directions until a set of 4 contiguous residues have an average of beta plated less than 100. You can move to the beta sheets until the accumulation of the values is less than 100. If it is more than 100, you will consider it in beta sheets, and if it is less than 100, then the beta sheet will finish. So here we have the table. This table, you have to understand this table, and I hope other students will understand it from YouTube, because I don't know why your class always behave like so immature. So here we have a table. Kashif Slim, please be focused. If you will understand this table,
Yes,
it's a very easy concept of 2 to 3 minutes, and if you guys will understand it, you will be in a good benefit regarding secondary structure prediction. This is a peptide SPA, SEA, SDGQ, SV, SV. Okay, so here we have
yes.
PA mean propensity of alpha helix, and PB mean
Frequency of beta sheets.
beta sheets. And he just wrote the values, like serine is 77 value and for alpha helix, and for beta sheet it's 75.
75.
Proline 55, 55, alanine 142 and 83, serine 77, and here we have no value. From where we derived these values? From this table 7.1, the Chou Fasman parameters for the 20 common amino acids. This table is from the paper they published in actually. So here you can see they have 20 amino acids from alanine. In alanine they have the propensity value 142, then 83, then 66, and all these values. You can see the values, man.
Yes, sir.
Okay, from lysine we have 114, 74, and 101. For serine we have 77, 75, 143. For valine 106, 170, and 50. So you have to wrote the values from this table in the sequence of the peptide, and then you have to accumulate the values or add the values. Like if serine, proline, alanine, and serine, we combine these values 77, 55, 142, and 77. It's almost, you can say, more than 400, 350 or 400, right? And
Yes, sir.
from the
370,
previous,
I think.
yes, 370 somewhere. So from the last concept we just discussed that if the value is greater than 100, accumulation of 3 amino acid values greater than 100, we will consider it as a beta sheet.
Beta sheet.
So this is the main concept. Now we have another peptide SPAS EASDGQ EFETTY. So here you can see 1, 2, 3, 4, 5, 6 amino acids, and these 6 amino acids are in blue color. So here we have the
Yes, sir.
propensities of these 6 amino acids, and we can calculate. So here we have the formula, like first one, 4 of 6 amino acids have the propensity of alpha greater than 100. You can see here, if you will accumulate or add the values of 4 amino acids, it will be more than 100. Extend right until 4
400, sir.
contagious amino acids. So this from here you can move to the right direction, and then you also have to move to the left direction to calculate, as we discussed in the previous slides. Residues have p propensity of alpha is smaller than 100, then you have to calculate these values like mentioned here. Okay, so we just don't sum all the values because it will be complex for you. In actual backend Chou Fasman, there is a software Chou Fasman that performed all these experiments. So everyone in the class should have to solve this query. Sorry, this query. You can see, Kashif Slim,
Yes, sir.
find potential alpha helix. From this
Yes.
peptide, you guys have to
Under
calculate the only alpha helix by
under the
using these
table,
values.
sir.
Yes, under the table. You have to use this table and the values from the table and calculate. Okay. Then we have a conclusion,
Yes.
accuracy of Chou Fasman predictions. It's good one for two-dimensional structure prediction. Sequence having known three-dimensional structure are processed so that each residue is assigned to a given secondary structure class by looking at the backbone angle. We discussed in detail from where we drive everything. Three classes mostly we have: alpha helix, beta pleated sheets, and turns. I think there is some mistake. It should be B and T, or maybe E and C for denoting. And then we have
Yes, sir.
alpha helix, beta pleated sheets, turns, and loops. Okay, we will discuss. जी काशिफ सलीम.
सर ये टेबल हमें अवेलेबल होगा या हमें ये सर याद करना पड़ेगा?
नहीं, ये अवेलेबल होगा और आपको ये याद करने की जरूरत नहीं है. आपको सिर्फ कंसेप्ट क्लियर करना है, हाउ दिस टूल वर्क्स बैक एंड. ये आप कहीं पे भी कैलकुलेट लाइफ में नहीं करोगे ये, लेकिन आपको ये पता होना चाहिए कि टूल बैक एंड पे कैसे कैलकुलेट कर रहा है सेकेंडरी स्ट्रक्चर के लिए.
इज इट जस्ट अ मैकेनिज्म फॉर टूल, सर?
जी बिल्कुल, ये चाउ फैसमैन का जो एल्गोरिथम है वो समझाया है, और ये चाउ फैसमैन किस एल्गोरिथम और किस वैल्यूज की बिना पर ये कैलकुलेट करता है. और कंटिन्यूअस जो अमाइनो एसिड्स हैं जो एक दूसरे के साथ आई जा रहे हैं, ठीक है, जैसे ए पी आ रही है ना, ये एक दूसरे के साथ आ रही है, इस सेंस में उसने कंटिन्यूअस का है. और आप ये भी कह सकते हैं नेबरिंग वाले. ठीक है,
यस सर.
तो ये आपको कैलकुलेट सिर्फ इसलिए करवाया जा रहा है ताकि आपके कंसेप्ट क्लियर हो जाएं इन एक्चुअल चाउ फैसमैन एल्गोरिथम क्या था और वो कैसे काम कर रहा है सेकेंडरी स्ट्रक्चर प्रेडिक्शन के लिए. ठीक है, तो टूल्स अवेलेबल है सेकेंडरी स्ट्रक्चर के लिए. आई टेसर बहुत अच्छा है. आई टेसर से जो सेकेंडरी स्ट्रक्चर आता है वो बड़ा रिलायबल होता है, तो टूल ये सब कर देगा. ये सिर्फ आपकी समझ के लिए बात हो रही है. ठीक है बेटा. सो दिस वाज आवर लेक्चर ऑफ टुडे दैट योर क्लास ruined badly. And
सर
जी बेटे.
सर इज दिस मोर एक्यूरेट देन आई टेजर?
Both are okay, depends on the nature of the protein. Usually in bioinformatics or in computational analysis, we use more than one tools for the reliability of the results. If you want reliable results, you have to. Yes, so that's the story.
Thank you, sir.
You can see my screen.
Yes, sir.
What is in the screen?
Sir, conclusion, sir.
Okay, conclusion. Stay with me. Okay, what you are seeing on my screen now?
Sir, it's CFSSP.
Yes, CFSSP, Chou and Fasman Secondary Structure Prediction Server. We have this server and citations are available. So I just Google it with this name and then I just opened it. Now I am just going on Uniprot. Uniprot. Okay, what is on your screen from my side? The same software?
Yes, no, sir.
Uniprot.
Second tab.
Okay.
On second tab, sir. Yes, sir. Uniprot.
Okay. Now I am just going to take one sequence from Uniprot of protein. Like I just choose a DAOA protein, schizophrenia protein, and just going to take, it has 4 isoforms, and I just take a FASTA format sequence and paste in, sorry, why it's not, okay, here. and now just click on the predict button. Now you have
Okay,
the
sir.
secondary structure. Here we know, in red color we have alpha helices, in black color we have beta sheets. Like this protein has 153 amino acid from 0 to 153. So the secondary structure
Yes.
is like, suppose you can say till this amino acid almost 10 amino acids or 8 amino acid alpha helix. Then you have beta plated sheet. Then in yellow color you have coils, and in blue color you have turns. The same is
Yes.
mentioned here, the secondary structure of the protein, and then total residues and everything. This tool you can
Yes.
use for secondary structure prediction. You can copy the results, you can analyze the results. And yes, you have a question, please.
Sir, is this tool used only for secondary structure prediction?
Yes, this tool is for secondary structure prediction. So,
Okay,
but
sir. Thank
there
you.
are so many tools available for secondary structure predictions. I think we discussed in our class or not, I don't remember. Please recall me.
Yes, sir. We discussed already.
Yes, there are so many tools. So in your course content we have this Chou Fasman algorithm. So that's why I explained this for you guys.
Okay, sir. Sir, I use iTaser. So can I use on place of that?
Yes, you can, but iTaser is also good one. iTaser is reliable one too. You can use both one.
Okay.
You can use
Okay, sir.
both. You
Thank you.
can compare the results, and I hope the 80 to 90% or maybe 100% you will get the same results if both the tools are working on the same algorithm. I don't remember the algorithm of iTaser, but the Chou Fasman, this tool is just working on the algorithm we just discussed, and the values were derived from a solved structure of X-ray crystallography and NMR. Okay,
Okay, sir. Okay,
please
sir.
mute your mic. Thank you to be with me. Okay, Aisha
Thank you,
Hafiz,
sir.
please unmute your mic.
Yes, sir.
बेटा समझ आई आपको लेक्चर की?
Yes, sir. एक बार और देख लेंगे तो अच्छे से आ जाएगी.
चलें, very good बेटा. And okay, please mute your mic. Mohammad Imran, unmute your mic.
Yes, sir.
Did you get the points? What is the propensity? What is the propensity of alpha helix? How we calculate the propensities, from where the table derived? Did you get all the points?
Ji, sir.
Very good. Did you know how we run this software, Chou Fasman secondary structure prediction software, and we have the results?
Yes, sir.
Very good. Mohammad Mazhar, please unmute your mic. Everyone, please mute your mic except Mohammad Mazhar. Walaikum salam. So can you please interpret the results? It's on your screen, 0 to 153 amino acids. Can you please help me?
यस सर.
Please.
यस सर, इसके अंदर सर जो रेड कलर में है सर वो हेलिक्स है, और जो
जी.
सर ग्रीन कलर में वो शीट्स हैं, और जो इसके अंदर सर ये पर्पल कलर में एक लाइन नजर आ रही है सर टर्न है, और दूसरा ये येलो कलर में वो कॉइल है सर, और ये इसकी जो टोटल लेंथ है 153 है.
Very good, very good. Okay,
और
Mahtab
सर
Ali.
इसके
Yeah, जी बेटा, जी, sorry, जी.
सर कुछ क्वेश्चंस हैं सर, वो आपसे पूछने थे. तो सर
Sure
इसके अंदर
बेटा.
आप बता रहे हैं कि एक्सरे क्रिस्टलोग्राफी और एनएमआर की आप उसके अकॉर्डिंग इसको वो जो सर रेजिड्यूज हैं इसके जो रिजल्ट्स आते हैं, अच्छा इसके
जी,
अंदर
जी.
कुछ डिफरेंस आता है?
किस सेंस में?
मीन के एवरी अमीनो एसिड जो पे हम रिलाई करते हैं सर, तो
जी, जी.
इसके अंदर जो टेक्निक्स यूज होती हैं सर एक्सरे क्रिस्टलोग्राफी और एनएमआर,
जी,
सर
जी.
इसके अंदर जो रिजल्ट्स हैं, चाहे इसके जो 2D प्रेडिक्शन में रिजल्ट्स में तो कोई ना कोई इसके अंदर को मीन के सर रिजल्ट्स में डिफरेंस आता है सर, या मोर एक्यूरेट दोनों है, या इन दोनों में से कोई एक हो सकता है?
किन दोनों में से? एक्सरे और एनएमआर
मीन
में से? बेटा कुछ स्ट्रक्चर्स होते हैं वो एनएमआर से प्रिडिक्ट होते हैं, कुछ एक्सरे से होते हैं. अब सपोज करें प्रोटीन ए जो है वो सिर्फ एनएमआर से निकली है, प्रोटीन बी जो है वो एक्सरे से हुई है, तो आप उस हिसाब से वैल्यूज ले लोगे. बहुत कम प्रोटीन्स होती हैं जो दोनों टेक्निक से हुई हों, तो फिर उस वाली को देखा जाता है जो ज्यादा लेटेस्ट स्ट्रक्चर हो और जिसकी वैल्यूज अच्छी हों, फिर उसको कंसिडर किया जाता है.
लास्ट क्वेश्चन सर, एक
जरूर बेटा,
सर
जरूर.
मैं इस स्टार्ट में सर आपने बताया था कि ये एम्पिरिकल एल्गोरिथम है सर, व्हाई वी यूज एम्पिरिकल एल्गोरिथम इन दिस सर्वर?
ये एल्गोरिथम को उसने एम्पिरिकल एल्गोरिथम का है जो कि एक्चुअली डिजाइन किया था चाउ एंड फैसमैन ने. अगर आपने एल्गोरिथम्स को पढ़ा है तो देयर आर डिफरेंट टाइप्स ऑफ एल्गोरिथम्स. वो
Yes,
जस्ट एल्गोरिथम को स्पेसिफाई किया, उसने स्पेसिफिकली एम्पिरिकल एल्गोरिथम नहीं इस्तेमाल किया। ये एल्गोरिथम बनाया था चाउ एंड फैसमैन ने, और ये
yes,
जाके जैसे आप बात करें, अगर सपोज आप बात करें बाइक की, कि मैंने बाइक ली है,
Yeah.
तो होंडा एक कंपनी है, आपने उस कंपनी की बाइक ली है, ठीक है। होप यू गॉट द आंसर।
चलो सर, ज़्यादा
एनी अदर क्वेश्चन?
नो सर।
थैंक यू। महताब अली, प्लीज
Exactly.
अनम्यूट योर माइक। बेटा, व्हाट इज दिस स्टेरिक डिनोट्स ऑन योर स्क्रीन? आई जस्ट हाइलाइटेड।
ये स्टैंडर्ड स्ट्रक्चर के नीचे है सर, ये क्यूरी 1 हेलिक्स 1 इससे ऊपर
नहीं
से
नहीं, ये
static
जो है बेटा स्टेरिक, ये क्या बता रहा है?
सर ये
उसामा उसामा मुनीर,
जी सर।
बेटा ये आपको
आवाज आ
पता
रही
है?
है सर?
जी बेटा, आपको पता है ये स्टेरिक क्या
सर
बता
ये
रहा है?
आई सर ये टर्म्स तो नहीं बता रहा,
जी, आप बहुत
जो
करीब हैं,
टर्म्स आ रही हैं।
आप बिल्कुल बेटा बिल्कुल करीब करीब हैं कि ये बता रहा है। बताएं, आप तो रुक ही गए हो।
सर टर्म्स ही हैं
चलो जी, आप
सर,
लोगों के लिए एक
स्ट्रक्चर
क्वेश्चन
के
है।
बुनियादी टर्म।
एक क्वेश्चन मैंने आपको पहले
जी
दिया है
सर।
कि वो आपने सॉल्व करना है। दूसरा क्वेश्चन ये है कि आप नेक्स्ट लेक्चर में हम डिस्कस करेंगे कि व्हाट दिस स्टेरिक मीन। ओके,
ओके सर।
थैंक यू एवरीवन फॉर ज्वाइनिंग, थैंक यू सो मच। यस, काशिफ सलीम जी बेटा।
सर काइंडली रिक्वेस्ट यह है कि सर आप ऑनलाइन क्लास लेते रहें तो हमें सर इससे थोड़ा सा बेनिफिट हो जाता है कि आपसे क्वेश्चन वगैरह पूछ लेते हैं। और थैंक यू।
ठीक है, सो thank you so much for everyone for joining, and
ठीक है
I
सर।
hope what we learned we will save in our mind, and if anything wrong we learn, may Almighty help us to delete those
आमीन।
from our mind. Be safe from this pandemic. Please protect yourself and your families. Thank you so much.
थैंक यू सर। अल्लाह हाफिज़।
Allah Hafiz.